Greetings. My name is Morgan Mote, and I am accompanied in this presentation by my partners Rajiv and Chanh. I'm the leader for our bioengineering senior design team and make sure that the progress of our team is closely aligned with our advisor, Dr. Justin Jaworski's expectations. Our project is in collaboration with an electrical engineering senior design team, and they are advised by Dr. David Wetz, and we owe the electronic component system to their work. For this short lecture, we're going to be getting into the function, design, and description of how to use our 3D printer for fabricating protein structures with micron resolution. 3D printing has facilitated many scientific innovations over the years. It allows for the rapid prototyping of functional designs. The biomedical potential of this technology is generating a lot of interest. However, commercial bioprinting currently relies only on off-the-shelf polymers rather than actual biological materials, and the need for improvement is outweighing the current benefits that technology has available. As a result, there is a growing need to integrate 3D printing with formable biological materials. The technology we have contributed to is valuable in terms of tissue engineering because it will facilitate the generation of micron-scale collagen networks that help direct cell growth. Below, we can see one live cellular staining and 3 fluorescent staining images of collagen cells. The reason it's important to develop this technology is because only biologically derived protein networks, such as collagen, are able to provide the biomechanical and biochemical cues to direct cellular growth in living tissues. The photo to the right, we can see the hierarchy of collagen, and the images below, we can see different 3D matrix structures that are also made of collagen. Our low-cost 3D bioprinter incorporates a moving printing stage in conjunction with a micron diameter glass capillary filled with type 2 collagen that is extruded through this apparatus onto the printing stage. The system is capable of facilitating the rapid prototyping and custom 3D biomaterial scaffolding. It will be developed using 3 integrated mechanical systems. The speed, direction, and accuracy of the printing stage movement in conjunction with the capillary will be assessed via stereo microscope and the boundary detection software whose code is originated in Python. The electrical engineering team is responsible for the creation of the electronic control portion of this project, while we were responsible for the mechanical stage portion and the biochemistry aspects of the collagen extrusion mechanism. As we can see, the images to the right give a good view of the different components of the mechanical stage assembly that our team was responsible for. And when it comes to the bioprinter electronics, the microcontroller dictates the functions of the stage sensor and LCD and sends a pulse to the step motor by using a timer interrupt. We can see that the images below, starting with the one on the left, the stereo microscope view of the printing capillary aligned with the glass printing stage. In the middle picture, we can see the control functions of the joystick. The image to the right, we can see the speed settings on the LCD.
So in this picture, we can see 2 comparative algorithms. The one to the left is the simplified logic, and the one to the right is the specific electronic components that are controlled at each point. Understanding our way around this portion is very integral for us to be able to successfully integrate the mechanical stages with its components. Okay, in this slide, we get a good view of the logic circuit, and it is imperative for us to understand what this diagram means so that we can properly disassemble and reassemble the device. Our team's design process started off on a paper. Essentially, our first step was to perform the mathematical calculations in order to verify the part dimensions with considerations to the material properties of delrin that we are going to align with our project requirements. Next, it was to verify these dimensions in SolidWorks and make sure there are no virtual build errors. Next, we got the design 3D printed in ABS in order to physically confirm the dimensions in the lab so that we can put together parts with the corresponding screws and make sure everything fits properly. Once we are able to confirm sufficient tolerances and that the parts fit together as expected, the parts are then fabricated and built out of delrin. Once a delrin model has been produced, we assemble the device and evaluate direction, extensive vibration, speed of the movement, and the accuracy of the origin and the boundary detection software using stereo microscope, and then finally analyze the structures using imaging. Here is an example of the mathematical calculations we went through in order to determine the tolerance and resolution of our 3D printer. Okay, in this slide, we get a better view of each of the individual mechanical printing stages as well as the stage components.
On this slide, we can see that the purchased components, such as motors, lead screws, and support rods, were integrated with 3D printed polymer versions of the device to ensure proper integration of the baseplate and electronic components. As you can see in the image below the text, this is our disassembled ABS prototype, and then the images to the right, we can see a few different baseplate components and how we ensure they fit together. To give a good visual of the way our 3D bioprinter works, it is essential to understand how the speed of the printing stage determines the collagen structure and matrix shape. In our case, the volumetric printing area is maintained by utilizing the origin and boundary detection software that was designed by the electrical engineering team. This system is integral in the proper function and output of our device. We can see that the photoelectric sensor that's placed on the motor works when its signal is disrupted. Essentially, the sensor has a transmitter that emits a beam of light to a receiver, and when this beam is disrupted, the sender sends an output of 0 volts to the microcontroller, indicating that a boundary has been hit. Once disrupted, the sensor sends the information to the microcontroller to stop the motor shaft once the boundaries are hit. This feature can also be utilized in the same way to tell the user where the origin is in space. After a very complicated boot up sequence that only our advisor has access to due to safety and liability concerns, we are able to successfully utilize the 3D printer and control it using the PlayStation 2 controller joystick. Once speed settings have been made and the origin has been detected, collagen can then be extruded out of the capillary and onto the glass printing stage as shown in the middle photo outlined in red. The photo to the left demonstrates the ready state of this device, and the photo to the right shows the Python code of the stereo microscope stage view in the ready state. We must make note that the current design shown in this photo is the Mach 1 mechanical stage design. Our third generation design is still being fabricated. However, the developments in the operation and the sequencing of this device have been made through this semester, and we successfully worked with the electrical engineering team in order to become educated users of this technology. Here are the most noteworthy references that we utilized for the transformation of our project. And finally, our senior design team has many people to acknowledge for the successful facilitation of our project. We would like to thank our advisor, Dr. Justin Jaworski, for project guidance and budgetary contributions. Additionally, we have the electrical engineering team, Dr. Wetz, Dr. Rebahani, Morgan Chivers, Samuel Law, Braden Itz, and Rami Barakat. Thank you all for your time, and we look forward to your feedback regarding our project.